talking so far about derivatives from the ectoderm, and today we're going to continue that theme with sort of a special case. We're going to talk about neural crest cells. And some people actually think of neural crest cells as almost like a fourth germ layer. And that's because they're so extensive and so proliferative throughout the body. They give rise to so many different types of tissues that they're really a force to reckon with in their own right. So these cells, as you may remember, are originating in the body just after neurulation, so just after we form the neural tube. If you look dorsal to that neural tube, sort of in between the neural tube and the overlying ectoderm, you can find this population of cells that are more or less individual cells just located in that position, and these are the neural crest cells. So this slide shows some of the things that eventually they can become in the body. One, we've already heard about pigment cells, so they're going to be migrating and creating the melanocytes that will integrate into the skin. We'll talk a little bit more about that today. Another big thing for neural crest cells to become is to contribute to some of the sensory neurons in glia. So this is a portion of the neuronal cells of glia are neural crest derived. Others of them may come from the neural tube directly, and so that's more of a direct ectoderm or neuroectoderm origin. But some of the neural crest cells can give rise to additional types of neurons and glia. Then this is the adrenal gland, so that's an organ within the body, and you can find some examples where neural crest cells contribute to some of the cells of organs and help them to function in their final and complete state. Connective tissue is sort of a general term for almost like filler tissue, basically tissue within the internal regions of the body that help things stick together. Neural crest cells can contribute to that. Cartilage and bone. So in fact, when we talk a little bit more about bone development, you'll see there's a whole category of bones within the body that have extensive contribution from neural crest cells. So this is actually an essential thing that neural crest cells need to do as development is proceeding here. And then neurons and glia, the cranial ganglia, so that's still within the same category of neurons, but I guess they wanted to highlight those for the heads here. So head tissues and trunk tissues, both having extensive neural crest contribution. So this is just reflecting the origin of these cells as the events that are going to give rise to neurulation begin to form. We're eventually going to end up with these neural crest cells here. In this picture, they're colored sort of light green, and that reflects, if we track back where they came from, two areas sort of at the borders of the neuroectoderm in kind of a greenish color in this figure. This is the site where the progenitors to the neural crest cells would have originally come from. So they're going to end up here as the neural tube is forming right on these ridges. And in this location, they'll already be expressing particular unique transcription factors. So you don't need to memorize these, but just as an example, Fox, D3, or slug are two things that are unique to these cells. So this paradigm of differential gene transcription in the process of a cell fate that's unfolding, that's already started to become evident at a really, really early stage here, where these cells are expressing unique markers. In this location, they are also subject to unique impacts from paracrine factors. So BMPs in the neural tube, when to the ectoderm, there's a region of intersection between these two that may be some of the important cues or inductive events for first identifying this group of cells. Okay, so then as neurulation proceeds, and the neural plate is beginning to buckle inward to form the neural groove, we have these cells that are going to become neural crest cells right at the ridges there. These cells are going to have to change their organization within the embryo. So at first, when the ectoderm was present, neuroectoderm was present, these cells were all in an epithelium. So they were in a layer together. They expressed cell junctions that might have helped those cells adhere to each other. And now that's all going to have to change, where these dark blue cells are letting go of their neighboring cells and becoming more individual and migratory. So this involves several things. They have to change their cell shape. They have to lose their adhesion to neighbors. They may have to increase their affinity for other things out here in the environment. So substrates like extracellular matrix or additional types of neighbor cells become more attractive to them. And they want to then move towards those regions and away from being part of this epithelium. So this is a critical step in the formation of neurocrest cells. But just in general, this business of going from epithelial to mesenchyme and or back again, mesenchymal back to epithelial, that's something we see a lot of now that the germ layers are going to start making organs or different types of tissues in the body. So keep your eye out for that kind of transition, because there are many of them in developmental biology, and cells can do this frequently in the course of their process of building an organ or structure for the body. Okay, so no surprise that neurocrest cells are highly pluripotent. And so this reflects their ability to give rise to daughter cells that have lots of different cell fates. So how do the neurocrest cells decide which type of daughter cell at which time and at which location? And this is one of the intriguing questions about neurocrest cells, and it genders a lot of interest for trying to understand how they make these decisions. And so one way to study this is simply just to be able to identify and track these cells as development is proceeding. So we know where they start out, even back to those green cells that were then underwent the EMT. And now from that point, if we want to track them further, one thing we could do is just to label them with a dye. So in this experiment, they've gone in here with a needle on either side of the neural tube, just injected some of those cells with a fluorescent dye that's not going to hurt them in any way, but will just allow us to track where they go as time proceeds in this embryo. And they go all over the place. So here they are growing down to some of these nerves and ganglia that are starting. Here they've become much more ventral in the organism, where the adrenal medulla in the adrenal gland is going to start to form. So they really migrate a long way, pretty much as individual cells, and then give rise to tissues down the line. So as they go, they may be subject to any kind of cues in their environment, juxtacrine cues from substrates that they're going to be finding, paracrine cues of things that were secreted depending on what they were passing through, what kinds of tissue they were passing through, and then just their own internal transcriptive factors as their own development and differentiation proceeds as time goes by. So I think all of these could be candidate signals for pushing the neurocrest cells to one or another eventual daughter cell fate. Okay, so here actually tracks some of the process of these multipotent or pluripotent precursors as they are going to make some decisions down the line. So initially, 4 different colors in this progenitor cell means that 4 different types of daughter cells could arise from it. And initially some decisions are made, perhaps based on location, that we're going to maintain the possibility for some daughter types and sort of not maintain other possibilities. And then all of these factors here are all paracrine factors. So we're going to see a lot of different external signals impacting these neurocrest cells as they're migrating around, and perhaps pushing them towards one or another daughter cell fate. So depending on the combinations of paracrine factors or local environments that they see, they could then differentiate to these various different types of daughter cells, which are quite different, but all derived from the neurocrest cells. So if we look around have the neural crest cell. The neural tube, of course, goes from complete anterior all the way down to the posterior. And neural crest cells are found along that entire location, and they're going to contribute to tissues all along the anterior posterior part of the body. So cranial tissues, cardiac tissues, trunk tissues, and then in a more minor sense, some of the things to do with the gut and sacral tissues. So we're going to just take a briefer look at three main types of neural crest cells, just to kind of give you a sense of what these cells are and what they're doing in these different locations. And they all give rise to many derivatives. This is sort of one of the themes for today, and here are some of the derivatives for each of these three main categories. So, in addition to nerves, cartilage, connective tissue, and many structures within the face actually have pretty extensive contribution from the neural crest. So, including teeth, ears, jaw, and then the pharyngeal arches, which were these kind of neck bars of tissue in the embryo early stages. All of these have a lot of contribution from neural crest cells. And then in the heart, in mammalian hearts, we actually form a couple of chambers, and then we have a wall that grows down in the middle of these chambers to separate them into two. So that's how we get two atria and two ventricles, is through the formation of a septum, or a wall that grows down in the heart as it forms. And those cells that contribute to that are in part derived from neural crest cells. So you absolutely need to have neural crest cells in order to get fully functional heart. And then also several tissues here within the trunk as well. So you don't need to memorize this list, but I like to put it up to give a sense of why these cells are interesting. So you might think, as a corollary to this group of cells being able to give rise to so many different types of daughter cells, that if anything goes wrong along the way, you're really setting up for a developmental disorder of some type. And so this is a little search that I did earlier this year in the online Mendelian Inheritance of Man database, which, if you haven't heard about this, it's kind of a cool thing to know about. What it is a database that is a collection of all that we know based on genetics and mutations, medicine, human disease, and biochemistry, kind of all collected in one spot. So you can go to this OMIM, Online Mendelian Inheritance of Man, and you can either type in a cell type, a gene, a type of disorder, and it will call up everything that we know about the genetic basis for that. Then if you click on one of these, so here's a syndrome called ABCD syndrome, I have no idea what it is, but you'll get a little synopsis from published papers in the literature saying, what do we know about the gene that is causing this? What do we know about specific human mutations? What do we know about the biochemical function of the gene? So it's really quite handy for any of you that might go on to graduate school or professional school. I'm sure would probably like to have a nice summary of this that's easy to access. So keep that in mind. Anyway, so if I type in neural crest, I got 107 actually quite different disorders or diseases that are related to the malfunctioning or the malpatterning of neural crest cells. So if these cells are not specified in the appropriate numbers, if they don't migrate to the right place, they don't differentiate correctly once they get there, you're going to have a problem in the tissues of the body that are derived from neural crest cells. So within the trunk, there are two main pathways that the neural crest cells are going to take. So let's have a look at those. So just to orient you here, we're looking at a cross-section of an embryo. You recognize the neural tube running anterior and posterior. Immediately below that is the notochord. And then on either side, we see two large masses of tissue. That's mesodermal tissue that's going to give rise to different territories of mesoderm. And then here's some more mesodermal tissue. And then the blue on the very outside, that's your ectoderm. It's covering the entire embryo, giving rise to skin, hair, and so forth. And one of the places that you'll find these neural crest cells, starting from dorsal to the neural tube, one of their roots is just below the ectoderm, kind of on the outside layer of mesoderm. So here they are. They're very dorsal, very lateral, and very superficial or towards the outside layers of the organism. So these cells are going to be migrating there between the epidermis, ectoderm derived, and dermis, mesoderm derived. So they'll be migrating along there. Here's a couple pictures. So these are neurocrest cells. They happen to be expressing a marker, which is a gene called KIT. And since they're expressing that and the neighboring cells are not expressing it, we can use that to detect these neurocrest cells, see where they're going, how fast, how many, what sort of dispersion pattern do they have. And in the wild-type situation, you can see here they are kind of migrating just as individual cells right through this region of the body. And these cells are going to become the melanoblasts. So as they migrate around, some of them will go up and start to colonize the overlying epidermis and then complete their differentiation as the pigment-producing cells that will give color to the skin and to the hair. So here they are, they're migrating. Here's an example of a mutant that happens to be in a transcription factor called MITF. And in this case, what we've done is just reduce the number of neurocrest cells that are migrating in this region. So occasionally you can see a lone guy there, but overall there's far fewer. And you're going to get fewer melanoblasts. And if you think about what might that look like, it's probably going to be an animal with either very, very pale skin or white hair color, basically lacking pigment because they don't have many of these melanoblasts. So they're expressing KIT here, and then also the red is another label that's MITF. So those cells are actually showing both of those gene products being expressed. And so what is KIT, just for interesting? KIT happens to be an important part of this signaling pathway here. It's a receptor. It, in fact, is a receptor tyrosine kinase, if you remember that from cell biology. So this is something where you can have an external paraffin factor ligand, which happens to be named steel. And in this picture, you see those two little yellow balls. Those are two steel paraffin factors bound together in a dimer. And they're going to interact with the KIT receptor. As a result of that interaction, now you see these KIT receptors become phosphorylated, and that activates them. Then they start this whole pathway of signal transduction, so they pass the information on through the cytoplasm, and eventually we'll get the MITF transcription factor in the nucleus to bind to target genes. And one of the target genes is tyrosinase, which happens to be the enzyme that's the precursor for both pigment proteins. Remember that, melanin and phenylalanine? So we need tyrosinase early in the biochemical pathway to make both of those kinds of pigments. So then if you look at mutants along the way, either a mutant in steel, in KIT, or in the MITF, you can see that the phenotypes you get are white, deaf. That's because neurocrest cells contribute to some bones of the ear, and so if they're not proliferating or migrating correctly, you don't get those bones forming correctly and you're deaf. Microthalamic, this means a small eye phenotype, so there's some contribution from neurocrest there as well, and then some immunology type phenotypes as well. So this is basically just tying all this together in terms of the melanocytes, a signaling pathway, and then eventually the production of pigment within those melanocytes themselves. So mutations in the KIT gene can give you some interesting pigmentation phenotypes. And this was one of these scenarios where there was this sort of phenotype known in the human population, and it was suspected that a mutation in the KIT gene might cause this. So now, this might not completely eliminate the melanoblasts, but perhaps just reduces their number or reduces their migration. So if you think about melanoblasts starting out on a dorsal side of an embryo and they have to migrate down all the way around to the ventral side, you know, there may be a little patch of cells where, or patch of the embryo where those cells never quite make it. So just that patch then lacks the melanoblasts and is pale in color. So then it turned out that when people finally got the mouse mutant lacking KIT, it has really the exact same phenotype. So that's sort of supporting this idea that the problem here, the mechanism behind this phenotype, is that the neural crest cells that should give rise to melanoblasts are either fewer in number or not migrating the complete distance over the embryo surface.
Is it specific to like those regions? Because it looks like on both like the baby and the mouse, like it's missing some on the abdomen and all that.
So this particular mutation is specific to those regions. And I guess what it's really telling us is that it has a very uniform or predictable phenotype from this particular allele of this particular gene. I'll show you a couple of other examples that in principle are the same, but they're different genes. But they're all getting back to the migration and the number of these neural crest cells and is that wild type or not. But yeah, this one is just really recognizable because of that forehead plus belly hyperpigmentation pattern. So kind of a nice example of where mice and humans sort of reinforce each other, and it seems all to make sense for once. Okay, I have to throw in some cat pictures whenever I can, so here we go. Cat pictures of cats here with white tips of the feet, you know, very, very distal. Neural crest cells have to go a long way to get there. White on their belly. Or a much more extensive version of really the same thing, where these neural crest cells are migrating kind of in an uneven fashion and not getting all the way around to all the parts of the skin and hair. So there is a genetic basis for this, and it's called white spotting is the phenotype that you see. And this is just a gene that's needed for again, for the proliferation and migration of neural crest cells in proper numbers. And it could be somewhat more severe in phenotype or somewhat more mild in phenotype, and you can vary the cat coat color pattern this way. Okay, second pathway for neural crest in the trunk region is going to be much more interior or medial, so much more towards the middle of the body. And that's shown here by this blue arrow that's going down the side of the neural tube and then entering here into this chunk of mesoderm tissue. And so it will be going directly through a chunk of mesoderm, but it's more specific than that. It doesn't just go to any mesoderm, it goes through a particular region. And so let's look at what that region actually is. And we have not yet talked about mesoderm, so I need to give you a little preview slide, which you'll kind of see this again once we get to talk about mesoderm. But for now, what we have here is on the side of the neural tube, initially there are these large bars of tissue. And then these are going to get broken up into smaller, discrete chunks that almost look like balls or cubes of mesoderm tissue. And these are called somites. So here you can see an embryo with about 6 somites that have formed so far. And this is a phenomenon that happens in a very time-wise fashion. So it's going to start out, you'll get one somite forming, and then another half an hour later, here comes another one, and then another one, another one, another one, until you can get the characteristic number of somites for each species. So once we get the somites, so picture a spherical chunk of mesoderm tissue, this is going to further subdivide into different regions. And those regions are called dermamyotome and sclerotome. And the sclerotome is the part that's the closest to the interior center core of the embryo, so closest to the neural tube or these light pink cells in this particular picture here. So these sclerotome cells are just certain territory within the somite. They have typical things they give rise to. And dermamyotome is a slightly different territory, and it gives rise to somewhat different types of things. But in general, we're talking things like vertebrae and muscle tissue and other good mesoderm derivatives. So the neurocrest cells are going to head right through the sclerotome tissue. But even more specific than that, if you look at each individual somite, the neurocrest cells are only migrating through the anterior portion of that somite. So the somites are these repeated units, and in the anterior half of each repeated unit, now you'll see trunk neurocrest cells zooming through that territory. And so here, to demonstrate that scientifically, they've labeled the neurocrest cells with an antibody that will give rise to a colorimetric reaction, so immunohistochemistry. And these black bars are just showing you where the individual somites are located. And in each case, you see the neurocrest positive signal being found in the anterior portion, but not so much in the posterior portion of each somite. So they're quite picky about where they go. And here in the midline of the embryo, where these neurocrest cells started, if they happen to be aligned with an anterior somite, or they just can head straight down. But if they're over a posterior somite, they're going to have to migrate a distance and then head down. So this suggests that they're really sampling cues in their environment and choosing a very deliberate pathway based on those cues. This may involve positive cues or negative cues to help direct these cells in a pretty organized way. Here's another piece of evidence that looks at neurocrest cells that have been labeled with a dye and just taking different cross sections through the embryo at the level of an anterior portion of the somite. And you can see all these, again, pretty much individual, mesenchymally organized, so not in a sheet, but just individually migrating cells streaming down from the dorsal part of the embryo into more medial and intermediate trunk areas. But in the posterior slice, you're not seeing that at all. You're just seeing cells up there that are going to be migrating anterior, posterior, and then head down when they get to the appropriate spot. So then people would like to know what some of these cues are. And one way to try to figure out what the cues are is to look for gene products that are expressed perhaps only in the anterior region of somites or only in the posterior regions of somites. So here's a situation where the neurocrest cells, they're found in the anterior half, and here's a gene product, ephrin, that has a very particular expression pattern only in the posterior halves of somites. And it turns out that ephrin is actually a repulsive cue. So if the neurocrest cells are going along and they sample their environment and they encounter ephrin, they don't like it, and they'll migrate away from that. So it really is a repulsive cue. The cells within the trunk region, within the somite, are expressing the ephrin, and then the neurocrest cells are expressing a receptor to that, an EPH receptor. And that allows them to sense whether or not ephrin is present or not. And so these ephrins and their EPH receptors, this is sort of another famous signaling molecule within developmental biology that we see popping up with different tissues at different times. And what's kind of unusual about it is these are both gene products are localized within the cell surface, or at least are tethered to the cell surface, so it's a juxtaphrin interaction. You're not getting any paraffin factors being released that diffuse away. You actually have to have a one-to-one contact between the neurocrest cell and the substrate cell. But the unusual thing is that as a result of this interaction, it's actually a repulsive force. So we tend to intuitively think, oh yeah, if they're interacting, they're going to stick together and they're going to be attracted. But in this case, the signal is a negative one, and it will tell the neurocrest cell to move away from the side of that interaction with this receptor. So if you have then the ephrin receptors are expressed on the neural crest cells, and the ephrins in the scleroderm cells. This gives rise to the repulsive nature, and those neural crest cells will move away from that territory. So that's an example of a negative cue. There are several positive cues as well. And this is a little assay where they've taken basically a petri dish and put some extracellular molecules that you might expect to find in an environment of migrating neural crest cells. And they've sort of painted these factors in stripes. So the pluses are stripes where the factor is present, and the minuses are stripes where it's missing. And then they seeded some neural crest cells onto this plate and allowed them some time to migrate. And in general, you can see they're tending to migrate in streams, which are actually aligning with the positive stripes of protein expression within that plate. So in green here, these are 4 different extracellular macromolecules: fibronectin, laminin, tenascin, collagen. So these are all great big molecules that are found outside of the plasma membrane in the extracellular matrix that cells are creating. And these are things that the neural crest cells might actually encounter as they're trying to migrate their way through this mass of sclerotome cells. So positive cues and negative cues kind of make sense. Okay, next thing to consider is cranial neural crest. So up in the cranial region of the embryo at this time, we have some of these developing vesicles. We have forebrain, midbrain, and hindbrain, and then the 5 vesicles, whose names you all learned. And then here in the hindbrain, we have the rhombomeres. That was one of our test questions. What are the segments within the hindbrain called rhombomeres? And these are little territories that you can make out, and the neural crest cells are contributing extensively to all of these tissues. So here's a picture of the forebrain is sort of folded over in this section, but then we have midbrain and then hindbrain here.